Hello, and thank you for clicking on the link to know more about the project. You probably have heard about the central dogma of the molecular biology, which essentially explains that how the DNA inside the nucleus of the eukaryotic cells can be transcribed, into the RNA, and the RNA in terms of the messenger's RNA can just be, translated into the proteins, the working machine of the living cells. However, in this very organized orchestra, which is essentially the transformations from the copy of DNA into proteins, there are some non-coding RNA molecules, which this is called the microRNA, which essentially range from 17 to 25 nucleotides, and they do not have any prescriptions for any proteins, and that's why we just call them non-coding RNAs. They can essentially disrupt the mechanism of the protein synthesis in the ribosomes of the eukaryotic cells. They just do that by two ways. Either they just bind to the messenger's RNA and block the sequence of the nucleotides, which are essentials for the protein coding, or either because in terms of, the 3D dimensions which they have, they just block the attachment of the ribosomes into the RNA, the coding RNA for the protein synthesis. And essentially that's can be just thought of one of the identifying methods of the cancers, the existence of the microRNA. In this project, we are looking into two different mechanisms. The first thing is that we are looking at, we hypothesize, we call that a null hypothesis, that it says that essentially there is no relation between the microRNA existence into the cell and also the cancer. For example, you can think of it in that way, that imagine that in the null hypothesis, we said that depending if there is a cancer or not, there is a monotonous, the level of the microRNA into the eukaryotic cells. However, we can also think of another thing as an alternative hypothesis, and we say that with the null hypothesis, we say that the level of the microRNA, depending if there is a cancer, it's going to change. We say that, for example, if there is a cancer, we say that either we can just represent the existence of the cancer as, for example, imagine with an intercept, we have, let's say, with the positive slope, or we have with the negative slope, which shows that, the existence of the microRNAs in the cancer. Then we can think of, if there is a cancer, the level of the microRNA in the cell can either be increased, which is going to be this case we call that case A, or it's going to be decreased, which we call that case B. And that's the thing that we just call that alternative hypothesis. In this project, the thing that we do is that I'm just going to use a hierarchical Bayesian methods in order to see that if we have the null hypothesis or alternative hypothesis, and if there's an alternative hypothesis, the level of the microRNA in the cancer is going to increase or decrease. And at the end, we are just going to use several levels of, of the hierarchy, and I'm just going to use also model comparisons to see that which one is going to be the best indicators of the existence of the cancer as a diagnostic tool.